Good afternoon, and thank you for joining us for the October 2023 edition of Lunch a la Carte, a monthly series where we introduce you to one of the previous or current researchers from Coins for Alzheimer's Research Trust, and this month is no exception. We are excited about our presentation today. A couple of quick announcements. As a reminder, this event is held monthly. The dates are already put on the calendar. You can find that if you're a Rotarian by going to the DAC-DB calendar, District and Club Database, and selecting multi-district events. But the easiest way for you to find it is by simply going to the CART Fund website, which is cartfund.org, and click on calendar. All of the dates are there. You can register. You can find the Zoom link, which of course is the exact same Zoom link that we use every month. And we hope that you will continue to join us for these monthly informative sessions. Lots of activities going on the CART Fund. We want to say a special thank you and shout out to all of our recurring donors who have signed up to donate to the CART Fund at mycartfund.org. We are really grateful for your continued support and, of course, all of the volunteers that do a great job for us. At this time, I want to introduce past District Governor Tammy Mosteller, who is going to introduce our speaker for the day.
Okay, so we're going to introduce Dr. Paul Greer, and the title of his talk is going to be Identifying Potential New Therapeutics for Alzheimer's Disease. Dr. Greer attended Harvard College as an undergraduate, where he graduated magna cum laude. He did his doctoral work at Harvard Medical School in the laboratory of Michael Greenberg, where he investigated the molecular underpinning of autism spectrum disorders. He received his postdoctoral training at Harvard Medical School under Dr. Bob Data, where he uncovered a role for MS4A genes whose mutation is strongly linked to Alzheimer's disease. Dr. Greer opened his own laboratory at the University of Massachusetts Medical School in 2017, where he is investigating the etiology behind neurodegenerative disorders and seeking new ways to treat these diseases. Dr. Greer has received a number of Young Investigator Awards for his work, including a Cyril Scholar Award, Rita Allen Foundation Award, and an NIH DP2 Young Investigator Award. Help us welcome Dr. Paul Greer.
Thank you very much for the really nice introduction, and thank you for giving me the opportunity to share with you a little bit about the work that we're doing that's being supported by CART. We're incredibly grateful for the funding, and I think that what we're doing, I'm very excited about, and I hope to convince you that it's exciting as well. So, as you all know, Alzheimer's disease is a debilitating neurodegenerative disorder that robs patients of their cognitive faculties, their memories, and ultimately their lives. It's currently the 6th leading cause of death in the United States. And as we're getting increasingly better at treating a variety of other disorders, most notably cancer and coronary artery disease, the incidence of Alzheimer's disease is only continuing to rise as our population ages. Current estimates suggest that the number of Alzheimer's disease cases will triple within the next 30 years or so within the United States. And so this is a growing major health crisis for our country. Despite this, and despite tremendous effort from a huge number of investigators in both industry and academia, current therapeutic interventions for treating or preventing Alzheimer's disease are highly limited in both their number and efficacy. There are a few things that have recently emerged that I think show promise, but even those, I think we would all agree, aren't sufficient. They're not going to sort of cure this disease. And so we're really looking for new ways in which we can treat this disorder. And I think that hope for new therapeutic targets, historically, all of the or most of the research has focused on amyloid beta, and which there's a lot of evidence to suggest is important within Alzheimer's disease. Lots of companies are developing antibodies or other therapeutic strategies for limiting amyloid beta, and these have begun to show promise. But I think that, as I said, none of these is curing the disease completely. And I think that there's a need to look outside of amyloid beta to see whether we can either find an alternative approach or do something that we can do in combination with amyloid therapy that we could use to treat this disorder. So wonderful. Can we just go to the third slide, please? Thank you so much. So I think hope for new therapeutic targets has emerged thanks to this ongoing revolution in DNA sequencing technology through which we're all living, which has enabled human geneticists to take hundreds of thousands of individuals who have Alzheimer's disease and hundreds of thousands of individuals who are exactly the same, except for the fact that they don't develop Alzheimer's disease. So same age, same race, same gender, et cetera. And we can then ask, are there genetic differences between the people who develop Alzheimer's disease and the people who don't? And when scientists have done these studies, what they've found is that over the last 20 years or so, a relatively small number of genes whose mutation significantly changes the likelihood that you will go on to develop Alzheimer's disease. So if you have one of the mutations that I've listed here on this slide, you're either much more likely or much less likely to get Alzheimer's disease than someone who doesn't. And because these genes, we know their mutation affects your likelihood of developing Alzheimer's disease, I think that these represent really attractive new therapeutic targets to consider in thinking about new ways to treat this disease. So from this list of 20 or so genes, can you just advance the next slide, please? We were particularly interested in mutations in the MS4A family of genes. And I'm going to tell you why we're interested in these MS4A genes. So we really have two major reasons why we're interested in these genes. The first is that the genetic data linking mutation in MS4A genes with an altered risk of Alzheimer's disease is very, very strong. So at this point, there have been dozens upon dozens of independent studies that people have performed looking at mutation of MS4A genes, are looking for mutations in genes and the likelihood that an individual will get Alzheimer's disease. Excuse me. And essentially all of these studies across a variety of different ethnic populations have all found that mutation in MS4A genes significantly changes the likelihood that you will go on to develop this disease. In fact, I would say that current genetic data suggests that up to 10% of all Alzheimer's disease cases worldwide may be attributable to mutations in this disease. And this is a staggeringly large number when you consider that there are approximately 6 million Americans alone who have this disease. So we're talking about millions of people worldwide are getting Alzheimer's disease because of mutation in these genes. The next slide, please. And the second reason why we're really interested in these genes, the next one. Sorry, I have animation. Yeah, there we go. Is that unlike many of the other genes on this list that have been identified as mutations affecting Alzheimer's disease, where the function of those proteins is largely unknown, we had actually been studying MS4A genes in a different context, and we knew something about how they worked. And what we knew suggested they would be particularly attractive targets. So what we knew is that MS4A genes are actually expressed in a small population of sensory neurons in your nose. And they're responsible for allowing mice to detect smells, chemical smells that are present in their environment. So what happens is as a mouse is wandering around, it uses mostly its sense of smell for figuring out what's happening around us. We use mostly our sense of sight, but mice use mostly their sense of smell. And these proteins, these MS4A proteins, bind to these chemicals and then signal their presence to the animal. And the types of chemicals that they signal are actually predator-derived signals. So signals from wolves, ferrets, foxes, things like that cause mice to run away in an innate way so they don't get eaten. So you're saying, all right, Paul, you're telling me that there are these MS4A proteins that smell ferrets. What does this have to do with Alzheimer's disease and why should we care? And the reason we care is that because MS4A genes encode these proteins called chemoreceptors, these proteins that bind chemicals and signal their presence, those type of proteins have historically proven very, very druggable. It's been possible to design compounds that either activate or inactivate those compounds. And we can thus manipulate the signaling of those proteins. So these two observations, the first, that MS4A mutation really changes your likelihood that you will get Alzheimer's disease and is probably responsible for 10% of all Alzheimer's disease cases, coupled with the observation that they encode chemoreceptors, these proteins that we know we're going to be able to develop compounds that inhibit or activate them, suggests that these are really attractive potential therapeutic targets. But that's the problem is that when we started this is where we were. We didn't know anything else other than what I've told you. And it's hard to imagine how you're going to take what I've told you and develop a new therapy. We don't even know whether MS4A genes are good or bad, actually, within the context of Alzheimer's disease. And the reason for this is a little bit complicated, but it's important. So I'd like to tell you, try to explain it to you briefly. So typically within your genome, you have a gene, which is DNA, and that DNA gets turned into RNA, and then the RNA gets turned into proteins, and proteins are the actual, functional units that do stuff within your cells and within your body. Typically, if you had a mutation in an RNA, it would influence the protein in some way that you could predict. We know if we have a specific RNA sequence, it gets turned into a specific protein sequence. The problem is in mammals, such as humans, genes are much more complicated than that. And there are regions of genes that don't actually encode RNA and don't encode protein. And almost all of the mutations that have been identified in MS4A genes that change the likelihood of whether you get Alzheimer's disease occur in what are called these non-coding regions of the protein, of the gene. So we don't know what effect they have on the protein. And therefore, we don't know whether they're harmful or protective. And if you want to develop therapies that target MS4As, you have to know whether you should be activating them or inhibiting them. So the first question we sought to address was this: are MS4A genes good or bad within the context of Alzheimer's disease? If I could have the next slide, please. So the way that we did this to ask this question is it's very difficult to do experiments in people. As I told you, we don't know whether these manipulations in people are good or bad. And so we turned to a mouse model of Alzheimer's disease, which allows us to recapitulate many features of Alzheimer's disease in a mouse. For the first set of experiments I'm going to tell you, we use what are known as 5X FAD mice. It doesn't really matter what the details of these animals are, but this is a very typically used mouse model of Alzheimer's disease. These mice harbor 5 different human mutations that cause early onset Alzheimer's disease. And they recapitulate in mouse many features of Alzheimer's disease. So mice are less good at tests of learning and memory. They have behavioral deficits. They have a lot of the cellular pathology like amyloid plaques, which are hallmarks of Alzheimer's disease. And so many researchers across the world use these mice as a way of studying Alzheimer's disease in an experimentally manipulatable system. So what we did was we took these 5X FAD mice and we deleted MS4A genes from them. So we got rid of the MS4A genes altogether and asked, do phenotypes in these animals get better or worse? So I'd like to just tell you about one experiment that we have done to test this learning and memory. So this is what's known as a novel object recognition task. So in this experiment, what you do is you take a mouse out of its standard laboratory cage on some training day. You put it in a new cage and you give it 2 identical objects. In this case, I'm depicting these with these blue cylinders. The mouse plays with these objects for a period of time. It gets familiar with them. It spends time exploring with them. It learns what they are. You then take the mouse out of this cage, you put it back in its regular cage. And several days later, you come back and you give it you do a testing day. And what you do is you replace one of these familiar objects with a novel object, in this case, this green cube. And mice are like people. They prefer new and shiny objects. And so if the mouse remembers what it did on the previous training day, on the testing day, it will spend more time playing and exploring the novel object than it does the familiar object. And we can quantify this by just dividing the amount of time that the animal spends playing with the novel object by the amount of time that the animal spends playing with the familiar object. So if you can go to advance the slide, please. Thank you. So here, I'm showing you a quantification of what this looks like. So on the leftmost bars, I'm showing you a wild type mouse. So here, its novelty score on average, this is several mice, but the novelty score on average is greater than 1. It remembers what it did on the previous day. It preferentially interacts with the novel object. So when you divide the novel object time by the familiar object time, you get something greater than 1. By contrast, if you look at the orange bar, which is the Alzheimer's disease mouse model, here the animal has a novelty score of exactly 1. It literally doesn't remember anything that it did on the training day. It interacts with these 2 objects equivalently and doesn't remember what it did. If you could advance the slide, please. So now if we delete the MS4s, which are in this yellow bar here, even in the context of this mouse that should be developing Alzheimer's disease and shouldn't be able to perform this test, Now the animal looks like a wild type mouse. Its inability to remember is completely erased. It now is perfectly capable of remembering what it did before, and it preferentially interacts with a novel object. So it's important to note that this is an experiment that's done in a mouse and not a person. And while this is a very good model of Alzheimer's disease, it's by no means perfect. And so we thought it was really important that we look at other models of Alzheimer's disease, other mouse models of Alzheimer's disease, that do this in a completely independent, that mimic the disease in a completely independent way. So if you could advance the slide. Thank you. So for the next set of experiments, what we did was we took a different set of Alzheimer's disease mice, which are known as MAP301T, MAP-T301S, which are a different mouse model of Alzheimer's disease, which in this case, rather than leading to the accumulation of amyloid plaques, they lead to the hyperphosphorylation of tau, which is another sort of hallmark of Alzheimer's disease in people. Again, just like the 5XFAD mice, this mouse model of Alzheimer's disease also recapitulates many phenotypes within these animals. And what we see is something very similar. Again, if you have a wild type mouse on the left set of these bars, the mouse is able to do this test of learning and memory. If you give it Alzheimer's disease, these blue bars, this effect is completely loses its ability to remember. It has a novelty score of 1, which is like pure chance. It doesn't remember at all. But if we delete the MS4A genes from this mouse model of Alzheimer's disease, we completely rescue this phenotype. It turns out that this is true not just of this test of learning and memory, but in the interest of time, I'm not going to show you test upon test. But in every test of learning and memory that we perform on these animals, when we delete MS4A genes, we completely restore the ability of both of these mouse models of Alzheimer's disease to perform these tests of learning and memory. So this is really exciting. It suggests that if we delete these genes, we can, even though the mouse should get Alzheimer's disease, it doesn't. It suggests that it's a really good a potential approach. But we wanted to extend this beyond learning and memory. There are a lot of other phenotypes that are observed in Alzheimer's disease besides learning and memory. And so over the next few slides, I'd just like to take you through some of the other ones that we've done. Can you just hit the next advance button, please? Thank you. Okay, so what we wanted to do next was to do another test of behavior, which isn't learning and memory, and which doesn't require us to sort of to impose what we think a mouse should do upon itself. So in this prior test, I told you like the mouse should play with a new object, but like maybe a mouse doesn't really want to play with a new object. It's sort of like, who knows what the mouse wants to do? It can't talk to us. So here what we've done is we've taken advantage of automated machine learning-based behavioral setup developed by Bob Datta's lab at Harvard Medical School that uses a completely unbiased automated manner of assessing all the behaviors that a mouse is doing without any human intervention. So what we do in this experiment is we take a mouse, we put it in any chamber that you want. This is a Y maze, but it can be in anything. We then put a video camera above the chamber and we record the mouse as it's behaving over 30 minutes, say. This is a typical video camera that you would find in your child's Xbox. It's called a Kinect camera. It captures both the X-Y position of a mouse, so where it is within the chamber, and also captures its altitude, which you can see pseudo-colored in the red. That's the mouse going up in height. It's like rearing up. So then we record all the behaviors that a mouse does over 30 minutes, and we use machine learning, the computer, to say, what are all the behaviors that a mouse has exhibited? So did a mouse turn left? Did it turn right? Did it dart forward? Did it rear up? Did it scrunch itself? Whatever it did, the computer finds it. And it calculates, if you go to the next slide, all the times and the percentage of time that the mouse spent doing each of these behaviors. So then we can generate these plots. And I should mention that if anything is unclear, please interrupt me. I'm happy to try to answer questions about it. On this x-axis, you see a bunch of numbers, 2, 6, 1, 12, 20. These are just automated characterizations of the behavior. So the computer isn't saying turn left, it's just giving it a number, 2. 2 might be turn left, but it just gives it a number. So we can say all the behaviors that a mouse displayed over this experiment. And then the height of each of these bars, the y-axis, is how frequently this behavior occurred over the experiment. And so what we can do is in the wild type mouse, we plot this in black. We have a bunch of behaviors that the mouse does. They occur at some frequency. If we then look in orange at the Alzheimer's disease mouse, what you see is that on the left, there's the appearance of these so-called syllables, these behaviors that the mouse, it displays when it has Alzheimer's disease or mimics Alzheimer's disease that it doesn't ordinarily. And on the right, it loses a series of behaviors that it should display. So these points are down here at the bottom. What's really cool is if we delete MS4A genes, which are these gray bars, the mouse now no longer looks like an Alzheimer's disease mouse. It looks like a wild type mouse. We've sort of reverted all of these phenotypes again. And if you go to the next slide, this is true in Alzheimer's disease, a second Alzheimer's disease mouse model, this MAPT mouse that I told you about earlier, where the Alzheimer's disease is in blue. So in both of these cases, for all the behaviors, we can sort of get rid of the phenotypes that are usually observed when we get rid of these MS4A genes. This is great. So now we want to sort of dive in and figure out why is it that when you get rid of these weird olfactory receptors that cause mice to run away from ferrets, do we affect Alzheimer's disease? Like, what is the connection here? And can we understand what's happening? Because maybe that will facilitate the development of therapeutic strategies. So if you go to the next slide. So the first experiment we did is a little complicated, and I apologize in advance. I'll try my best to explain it. So because we had no idea, we had no really good guesses of how MS4A genes were influencing Alzheimer's disease, what we wanted to do is we wanted to look at the whole brain without any bias. And so what we did was this technique called single-cell RNA sequencing. And in this experiment, what we do is we take the brain from an animal, we take a post-mortem brain, we dissociate it into single cells, so we get individual cells. And then we do sequencing to identify all the genes that are expressed in each of those cells within the brain. And what we can do to make this data easier to visualize is we can project all of this information into 2 dimensions. So here I've shown you this plot, this graph. You won't be able to resolve individual points because there are 100,000 plus cells on this graph. But each point on this graph represents an individual cell that was sequenced. And cells or dots that are closer to one another in this 2-dimensional space are cells that have more similar, they express more similar genes, and thus they're more identical to one another. And so when we do this, we can see, are the cells from an Alzheimer's disease mouse the same or different than cells from a wild type mouse? So if you go to the next slide. So we can do this for all the different cell types within the brain. And we can ask, when you have a cell, is it close to a cell that came from the same type of mouse, or is it close to any of those other cells that are the same? So if we look at, say, a neuron within the brain, and you look at a neuron from an Alzheimer's disease mouse model, is it close to a cell from a wild type mouse by chance, or is it only close to cells that also come from an Alzheimer's disease mouse? So are those cells from an Alzheimer's disease mouse more similar or different? And what I hope you can see from this graph is that most of the types of cells in the brain, astrocytes, endothelial cells, neurons, oligodendrocytes, parasites, they're all sort of random. They are close to this dashed line, which means it's almost random what type of other mouse they're next to. The exception is these microglia, where in an Alzheimer's disease model, if you're a microglia, you're much more likely to be next to another microglia from an Alzheimer's disease mouse than, say, a microglia from a wild type mouse, or a microglia from an Alzheimer's disease mouse in which we've deleted MS4s. And this is really interesting to us because microglia are the resident immune cells of the brain. And scientists now think that microglia play a crucial, although still somewhat unclear, role in Alzheimer's disease pathogenesis. There's an increasingly large focus on the role of microglia within this disease. And so the fact that all the other cells are pretty much the same, but microglia are changing when we delete MS4s, and that these cells have been shown to be really important, suggested to us that we really should be focusing on microglia. If you go to the next slide. Thank you. And another reason why we thought this is if we look to see in which cells MS4s are expressed, they're expressed just in microglia. And you can see that most easily when you quantify the panel on the right. We don't detect any MS4s in neurons or astrocytes or oligodendrocytes or other cells within the brain. We just detect them in microglia. So now we know deleting MS4s improves phenotypes in mice. we know that the phenotypes, the cells that are changing are microglia. Microglia are really important in Alzheimer's disease, and MS4As are expressed within microglia. So together, this suggests, let's look at microglia and see if we can figure out what's going on. So if you can go to the next slide. Thank you. So here's again, another one of these single cell experiments, but this time I've zoomed in just on microglia. So what you can see is if we have this, the cells in red come from a wild type mouse. You see microglia are mostly on the left, the right, and the bottom. There are very few cells that are up here on the top. But if we do look at the Alzheimer's disease mouse model, which are in turquoise, there's a bunch of cells here at the top. We've had this new population of microglia, this new type of microglia that are appearing. And if we delete MS4s in these purple, those cells mostly go away. And you can see that best in the next slide, where we've quantified that. Oh, the next slide. Sorry. Where we've quantified this. These are a type of cell known as disease-associated microglia. And disease-associated microglia have been shown to appear in the brains of people with Alzheimer's disease and not sort of under normal conditions. They're also present in other neurodegenerative disorders, and we'll get to that in a second. But again, in these Alzheimer's disease model, we see the appearance of these disease-associated microglia that also are present in humans. When we delete MS4A6C, these cells completely go away. So again, we're getting complete reversion of these sort of harmful phenotypes when we're getting rid of MS4A genes within Alzheimer's disease. If we could go to the next slide, please. So then we're interested in looking at other sort of hallmarks of pathology within these models. So I'm going to go through these fairly quickly just so we don't get bogged down in the details. But one of the first things we looked at was the hyperphosphorylation of tau. Here, on the quantification, you can see in these MAPT mice, this mouse model of Alzheimer's disease, we see this huge increase in hyperphosphorylation of tau. In green, when we delete MS4s, there's a significant reduction in the hyperphosphorylation of tau. It doesn't completely go away, it's important to note, but it is significantly reduced. Even more importantly, if you go to the next slide, one of the most important characteristics of Alzheimer's disease is a loss of neuronal synapses. So synapses are how neurons send signals to one another. They're a physical site of connection between one neuron and another. And it's thought that these are the sites in where our memories are encoded, where we facilitate behaviors. And what happens in neurodegenerative disorders like Alzheimer's disease is you have a loss in the ability of your synapses to function properly. There's a decrease in the number of synapses, and your existing synapses often also don't function as well. So again, we looked in this mouse model, and not surprisingly, if we look at the Alzheimer's disease model, we see a reduction in the number of synapses, and this phenotype is completely rescued when we delete MS4. So this is really important because we think that this is an essential site where Alzheimer's disease phenotypes are occurring. Now, if you go to the next slide, so this is the previous slide that I showed you is the loss of neuronal synapses. And we've been talking about microglia, and now I'm throwing neurons in there. But why is it that we think that this is important? So one of the primary ways that synapses are eliminated during neurodegenerative processes are microglia actually eat them by a process called phagocytosis. A microglial cell will surround the synapse and it will gobble it up. And what we found is that if we delete MS4s, this process of microglia gobbling up these synapses is reduced. I'm just showing you a reduction in this in vivo, but we see this also in vitro. And so we have a lot of experiments now that have demonstrated that if we delete MS4A genes, we inhibit this process by which microglia phagocytose or eat up these synaptic connections, which we think are crucial for Alzheimer's disease, both for the degenerative processes, but also for your cognitive functioning as well. So together, all of these data that I've shown you suggest that microglia are important. MS4s are expressed in microglia. When we delete MS4s, we prevent microglia from having these harmful effects on neurons. And the net effect of this is that you get a restoration of cognitive functioning in mouse models of Alzheimer's disease. So this is fantastic. Can you go to the next slide, please? So we were at this point, and then I had a new student join the lab whose name was Kit Mokarski. And she was like, well, there are a lot of similarities between Alzheimer's disease and other neurodegenerative disorders. And she was particularly interested in one of them called ALS, or Lou Gehrig's disease. And she's like, well, if these MS4A genes are so important in Alzheimer's disease, maybe they're important in these other diseases as well. And so what she did was she took, thousands of people who have ALS, thousands of people who don't get ALS, and asked, does MS4A mutation, again, correlate with the likelihood that someone is going to get ALS? And what she found is that it does. If you mutate MS4s, you also change the likelihood that an individual will get ALS. And you also change how the disease occurs for someone who does get ALS. So if you have a mutation in MS4s, you prolong lifespan of someone with ALS. And so she went on and did similar experiments to what we did with the Alzheimer's disease. If you go to the next slide, please. She found that if she takes a mouse model of ALS, deletes MS4A genes, she can get these mice to live longer and to do better in motor functioning. So ALS is a motor neuron disease. You get elimination of motor neurons, and it impacts your ability to perform motor tasks. If she measures this rotor-rod assay, which basically you put a mouse on a rod, you make the rod spin around and measure how long the animal can stay on it before it falls off. If you do this in a mouse model of ALS, they fall right off immediately. If we delete MS4s, which are these blue bars, if we delete MS4s in these red bars, the mouse stays on for significantly longer. So it lives longer, it does better in learning and memory. The next slide, we also see impairment in motor neuron death when we delete MS4s, and an inhibition in this neuronal activation of these microglial cells. So both Alzheimer's disease and ALS, we see when we delete these genes, we get really significant improvement in phenotypes. And I just want to show one more disease. If you go to the next slide. So Jason Friedman, who is an MD PhD student in the lab, said, well, if this is true for ALS and Alzheimer's disease, is it true for other things? He's looking at Huntington's disease and multiple sclerosis. I'm only going to show multiple sclerosis data very briefly. And basically, he sees the same thing. If we induce a model of multiple sclerosis in mice, it's called EAE, and delete MS4A genes, we again see a very significant improvement in how these animals do in this disease as well. So in all the neurodegenerative disorders that we've looked at, if we delete these MS4A genes, inhibit their function, we see improvement in clinical phenotypes, extension in lifespan, improvement in motor functioning, and improvement in all of the pathology that we have examined. So together, this plus the human genetic data suggests that strategies inhibiting MS4As are really an attractive thing to think about, to try to pursue for developing therapies that we might actually be able to use in people. And so that's really the focus of what we're doing now, and the focus of the grant that we received from CART, which we're really grateful for, which is that we're looking for inhibitory strategies by which we can get rid of these genes or inhibit the function of these genes and look to see what happens. So we're taking two different approaches. I'm going to talk about them really briefly because I know we're coming up against how long it should be. So one strategy is what's known as small inhibitory RNAs. And the nice thing about this approach is twofold. One, that UMass Medical School, where my lab is located, is one of the world's leading centers at this technology. And so I get to take advantage of rich collaborations that I've developed with my colleagues and their expertise to develop siRNA, small inhibitory RNA approaches to inhibit MS4A genes. And so what we've done is we've taken MS4A genes, we've developed a huge number of these siRNA compounds, and we've tested their ability to inhibit MS4 function in vitro and in vivo at this point. And these approaches have led to the identification of 4 different siRNA compounds that are able to very, very, very efficiently inhibit MS4A expression over a protracted period of time in the brains of mice. And so what we've now done, I unfortunately can't tell you the results yet because the experiments are ongoing, but what we've now done is we've taken these siRNA compounds and we've put them into mouse models of both ALS and Alzheimer's disease. And we're asking, do we see similar things to what we have observed when we genetically delete these genes, when we do these siRNA compounds targeting MS4s, and do we see improvement in phenotypes? And if we do, then these are really attractive lead compounds for moving into human models of the disease. The second approach that we're taking is going all the way back to the beginning of this talk, where I told you one of the things that we really liked about these MS4A genes is that they're chemoreceptors, these proteins that signal the presence of extracellular chemicals. And the reason why we like that is because these compounds, you can develop or identify compounds that are able to inhibit their function. And so what we've done is we've taken advantage of an in vitro assay that we developed for activation of MS4A signaling. And we've screened a huge library of already FDA-approved compounds that are able to inhibit MS4s. And these approaches have led to the identification of compounds that are already FDA-approved that are able to inhibit MS4A activity in vitro at very low concentrations, at concentrations that you would be able to use therapeutically. And so what we're doing now is we're taking these compounds and doing the same thing. We're putting them into mice and asking, do they also have an effect? And if they do, these are really attractive lead compounds for thinking about therapeutics. So next slide is just basically summarizing this. So the idea is we're doing this in mouse models of multiple neurodegenerative disorders, and hopefully we're leading to new ways of treating these diseases, either on their own or in combination with other existing therapeutics. And lastly, I just would like to, the last slide, please, thank the people in the lab who contributed to this work. I'm really fortunate to have a really talented group of scientists in my laboratory, a subset of which are pictured in this picture. I don't have a current picture, so, but I'm really grateful to them, and I'm particularly grateful for our sources of funding, especially CART, for allowing us to pursue this line of research, which I think has a lot of potential. So thank you very much for your attention, and I'm happy to try to answer any questions you might have.
Some of the things I noticed was that you deleted the MSO4. Am I understanding that you deleted this SO4 through these siRNA compounds, or how exactly did you delete that MSO4? I'm all pain. MS4A, excuse me.
That has a silly name. I apologize for that. We didn't give it the name. So the MS4A stands for membrane spanning 4A. It's just the protein goes through the cell membrane 4 times. So they called it membrane spanning 4A family proteins. Really creative name. So in the mouse experiments that we did, the first set of experiments until the last 2 slides, the way that we did this is we genetically deleted it. So what you can do in mice, one of the really nice things about modeling these diseases in mice is a technology has been developed. There was a Nobel Prize that was awarded for this, where you can take any gene that you want and you can get rid of it in a mouse. And then you can say, what happens in this mouse when I get rid of this gene? So the mouse is completely the same as a regular mouse, except we've literally come in with scissors. We've taken out this gene. It doesn't exist anymore. And then the rest of it is all the same. So we did this.
How do you remove that gene though?
Yeah. So what we do is we use this technology called CRISPR. So what you can do is because the sequence of the mouse is known, we know what every single DNA molecule is within the whole animal. We can design with a CRISPR-Cas9 system where it recognizes a specific sequence. It like goes through all 3 billion bases and it says, oh, I'm looking just for this one sequence. There, I found this sequence. I'm going to make a cut. I found this other sequence. I'm going to make a cut there. I cut these 2 sequences, and whatever's left in the middle comes out, and these 2, the rest of it joins back together. There's cellular mechanisms by which it's allowed to do that. So what we did was we specifically targeted these MS4A genes because we knew the sequence around them. We got rid of them. All the rest of the genes are the same in the mouse except for this one gene, and then we can ask what happens.
Okay.
Oh, sorry.
No, go ahead.
The siRNA compounds that I'm talking about later, like at the end, so these were not genetically manipulating the mice. What you do there is you put in a small inhibitory RNA sequence. So it's a short RNA sequence that's complementary to the gene that you're interested in targeting. So in this case, the MS4A gene. So the MS4A gene is 1,000 nucleotides long. This siRNA is short. It's like 20 plus around 20 nucleotides. It binds to a little bit of it, And then it causes this gene to get chewed up, the RNA of the gene to be chewed up. So the gene is intact, but it's just its ability to make an RNA that then gets turned into a protein is inhibited. And so it's a way of not genetically manipulating the animal that you can target it. And that's, there are strategies that people are using where they're actually trying to do gene therapy, where they're manipulating the genome of an individual. But the nice thing about this is that you don't require any manipulation. This is just relying on RNA. And there's a lot of development recently on RNA therapeutics. So both siRNAs, they're like, if you've been vaccinated against COVID, those are RNA vaccines. There's a lot of these, antisense oligos, a lot of these strategies that are coming on, they're taking advantage of these technologies or related technologies to develop therapeutic approaches.
Okay. You also said that you've done these tests on these postmortem brains of the mouse models. Have you done any type of postmortem brain sequencing or whatever of humans? And also, how could you delete? Because I know what you were saying, the deleting the MSO4. MSO4, y'all, is morphine. Sorry, it just rolls off the tongue from all those years of nursing. I'll just say MS4. So how could you possibly delete that in a human brain as well? Have you looked at that postmortemly yet?
Yeah, so it's complicated. So we haven't done an experiment yet in a human brain. I think it's a really good experiment, and it's something that we would like to do. Technically, it's challenging for a variety of reasons. What we are doing is we are getting blood-derived cells from individuals who have specific mutations in MS4A proteins. So we now are studying what the effect of human mutations are on the function of MS4. So we've taken sequencing information from many, many, many people, identified mutations that are present in them, and then found what is the effect of those mutations on the function of MS4 proteins, which hasn't been done before. We can then go back to individuals, to humans who have these specific mutations, and ask, if we take cells that are related to these microglia, so we can take macrophages, which are peripheral immune cells, which are very similar to microglia, can we see some of these things that we were talking about in those cells? So that's the easiest way for us to do the experiment, because it doesn't require, we can just get blood from someone. It's very sort of non-invasive and easy to do. And we're in the process of doing those experiments. I will say that now that we have been able to identify what the effect of the human mutations is on the function of MS4 proteins, we can now take these same mutations and go back and say, what is the effect of this on Alzheimer's disease and other related phenotypes? And we're doing that as well.
Okay. I'm glad you brought that up about the synapses. So I know a lot of times when I'm doing my dementia talks to people, I explain the synapses is like a ditch. You're going down the highway and you got a ditch and you have to jump over the ditch and keep going. And so that's how the information travels in the brain. And so I know when you hit that synapse, you have to jump over it and keep going. I often describe that as a detour. You're traveling down the road and all of a sudden the information goes boop, you have to take a detour, and then you go boop, you have to take a detour. And that way, people can understand it a little better. But you're saying that these macrophages are eating the synapse. So are they basically making the ditch bigger to where the information can't jump across? Is that what you're trying to say?
Yes. So the way that I think about synapses is basically, you have your TV. If your TV is not plugged into the wall, there's no signal that comes out. You're not getting any electrical current. If you take your TV plug, you plug it into the wall, now the electrical current can come from wherever your source of electricity is into your TV. And the synapse you can basically think of as the same as that, the outlet where the connection is actually occurring. So the synapse is really you're passing electrical activity from one cell to another cell. When we get rid of the synapse, it's the same thing as unplugging your TV. So the signal cannot go from your electrical source into your TV. There's no signal that comes. So what the synapse is what the microglia is doing is basically eating your connection, chewing up your wire. There's no electricity. Your TV can't come on, and you can't watch the Patriots.
Poor Tiffany. Okay, and I had one more question. I'm so sorry, I'm dominating. Anybody else got a question? Anybody else got something they want to ask? Please do. Okay, so So you've determined that the MS4A is high in Alzheimer's, ALS, MS, Huntington's, all these neurodegenerative disorders. So when this protein starts increasing, how can do you know how to determine whether or not it's going to develop into which one of these diseases?
Yeah, so that's a really good question. So we don't know. So, why is it that sometimes you get Alzheimer's disease and sometimes you get ALS and sometimes you get multiple sclerosis? And those diseases, there are some similarities, but they're very, very, very different diseases, obviously. And I think there's not a lot of evidence, but this is what I think. These are just one part of this disease. The MS4s are not the whole story here. Like, we're seeing significant effects, but people see significant effects when they get rid of amyloid or they get rid of in ALS, SOD, or, I think that there are many, many, many different components that go into each of these disorders. And I think that part of the reason why it's been so difficult to develop therapies is that it seems unlikely that we're just going to be able to take one thing and manipulate it and all of a sudden be like, oh, we've completely solved everything. Now people can live forever and we win. Like, I think that that's not the way, unfortunately, that this is going to work. I think that there are many arms of these diseases that are dysregulated and they talk to each other. So I think that if you affect one, you might have a positive impact on the other arms, because I think that you basically get sort of like an angry situation where you get, something goes wrong and then the immune system gets mad and then that causes degeneration and that makes the immune system more mad. And you just sort of are building in sort of this feed forward. The reason why it's degenerative, you have this feed forward cycle that builds and builds and builds over years. And so now I've lost my train of thought because I've just been rambling.
So the MS4A is just one portion. So say, for example, like for the Alzheimer's, it may be you've got the MS4 plus the tau plus the beta amyloid plus, but over here in ALS, you may have the MS4 plus this, and then and that's 100%. What makes it. And then the MS4 is just the common denominator between all of them.
Thank you for saving me from my loss of memory. So basically that's what I think, that in each of these disorders there's some specific feature of the disease that gives it its characteristics, and then there are some common features, and I think the MS4s are one of these common features. There's this this interaction between the nervous system and the immune system, and the MS4s are right in the middle of this, in these neuroimmune microglial cells, and those cells are important in all of them, but how they respond and why they respond and where in the brain they respond is different from disease to disease. And we sort of still have to sort of figure that out and decipher why it is that you get one disease versus another disease.
So is your lab basically trying to find some type of treatment to where you can effectively delete this MS4A in Alzheimer's patients to restore them back? Is that basically what your lab is trying to do?
That is our holy grail. That is what we were trying to do. If we can figure out some way to delete these genes or inhibit these genes in people, my hope is that in and of itself, or maybe in combination with other, therapies that other labs are developing or other companies are developing, we can then use that to treat these diseases after, hopefully in the early stages, so that we can, but I don't think it's something that you would have to take when you're 2 years old to prevent getting Alzheimer's. Right. This is something you would.
And how far are you guys from actually doing postmortem human brain studies?
Postmortem human brains. I think that we're closer to than putting these into a living person and seeing what happens. Mm-hmm. So I think that we're working very hard and we're trying to get there as quickly as possible. We're all racing against the clock here. So, we take this seriously and we're trying to do it quickly.
Yeah, I think you need to find a cure, because my great grandmother had it, my grandmother had it, my mother had it. They've all passed on, and my younger sister, at the age of 48, was diagnosed with early onset. So we need to find a cure so that I do not get this.
Yes.
This is great stuff, Dr.
Yeah, it's an awful disease. It's awful for the person who gets it, and it's awful for their families who have to sort of see it come through. We really are. We're working as hard as we can to. We and many other labs are working as hard as we can to try to.
We actually have two other researchers there at Harvard with you that were awarded in 2022, Drs. Kim and Quinty. and they are looking at repurposing some of those already approved FDA drugs. So hopefully, we're going to have a huge impact coming from Harvard with all of you guys working on CART grants.
Yeah, great. We're really, really grateful for the funding. We really appreciate it.
I think this is very enlightening for all of us, and we greatly appreciate not only your time, but you dumbing it down for all of us as well, so that we can understand in some form or fashion what it is that you are working on so diligently. And you certainly give us all a lot of hope. So we appreciate that as well.
Thank you so much, and hopefully some of it was clear at least. So I appreciate you taking the time.